Hey guys, welcome back to another video. So in this video we'll continue from where we left in the previous video. So we talked about exonuclease and endonucleases, about DNA manipulating enzymes. So we'll move on from there on. So stay tuned and watch the video till the end. So on with this, so we'll talk about polymerases in this section. So talking about polymerases, which are DNA polymerases, are enzymes that synthesize a new strand of DNA complementary to an existing DNA or RNA template. So polymerases has a very simple function of just adding new strands. Alright, so polymerase has a simple activity of polymerization, which is a process of simply adding new strands of DNA which are complementary to the existing DNA or RNA template. Alright, so according to the sequence which are present, complementary, it synthesizes new strands. So therefore there are four different types of DNA polymerases. Alright, so moving on with the first polymerase, which is DNA polymerase 1, which is prepared from E. coli. Alright, and this enzyme attaches to a short single stranded region or neck in a mainly double stranded DNA molecule, and then synthesizes a completely new strand, degrading the existing strand as it proceeds. So this is a simple thing, so I'll read it out for again. So this enzyme DNA polymerase 1 attaches to a short single stranded region, alright, in a mainly double stranded DNA molecule, and then synthesizes the completely new strand, degrading the existing strand. So talking about this, let's take this example. This is let's take what you call a DNA sequence. So it attaches to a short single stranded region or nick. So let's say it attaches here, somewhat here. DNA polymerase attaches here, so it produces a nick. Alright, and thus these strands are readily produced or reproduced, which are missing from here. So in this you can see a lot of them new things are existing, nucleotides are replaced, and it leaves in remaining existing strand as it proceeds. Alright, so this is the new strand that it replaces, but it forms. So I'll produce a better example for you in the next slide. So if you are not able to follow this one, so let's just remember that it attaches to a double stranded DNA or a single stranded region, then it synthesizes a completely new strand as of its function, but it degrades the existing strand as it proceeds. Alright, so DNA polymerase 1 is therefore an example of an enzyme with dual activity. It also has a DNA polymerization and DNA degradation. As you know, polymerization is its function and degradation is only correlated with DNA polymerase 1, whereas where it degrades the existing strand. So moving on with this, so let's talk about cleaner fragment, which is very important whenever we are discussing about DNA polymerase 1. Alright, so talking about cleaner fragment, the polymerase and the nucleus activity of DNA polymerase 1 are controlled by different parts of the enzyme molecule. So as we know, it has two features of polymerization and nuclease activities, which is known as the degradation. Alright, so the nucleus activity is contained in the first 323 amino acids of the polypeptide. So the removal of the segment leaves a modified enzyme that retains the polymerase function but is unable to degrade. So it is very obvious that the first part, which contains the nucleus activity, is removed. So it is only left with polymerase function as of now, and thus it is unable to degrade cause of its removal. Alright, so now this modified version of DNA polymerase 1 which is left now is known as the clinofragment. Alright, so the synthesize a complementary DNA strand on a single stranded template, but it has no nucleus activity, thus it cannot continue the synthesis once the nick is filled. Yeah, alright, so this is very important, that once the nick is replaced with other strands, it cannot proceed further because the nucleus activity renders or provides the DNA polymerase 1 to degrade the existing strand so that it can proceed further. So absence of nucleus activity or the degradation thing does not allow it to move further. So the major application of these polymerases is in DNA sequencing. The DNA sequencing is a very complex thing. So DNA polymerase 1 is used in DNA sequencing. Alright, so as you know, this is the example. As you can see, the clinofragment. So this is the strand. Here we have DNA polymerase 1, so it is now filled, As you can see in this, the neck is filled and thus it cannot proceed because of the absence of the nucleus activity or degrading thing. So it cannot say once the neck is filled or the replaced or it is replaced by the new strands, it cannot move forward because it cannot leave the existing strand. Alright, so moving on with this. So talking about some more DNA polymerases. So which one? First one is the Taq DNA polymerase, which is a special type of DNA polymerase 1, which is very important. So these three polymerases are very important: Taq DNA polymerase, PFU polymerase, reverse transcriptase. Alright, so talking about DNA polymerase, it is a special type of DNA polymerase 1 and it is purified from Thermus aquaticus bacteria. Alright, and this organism lives in hot spring. This is a very important point. And this enzyme is very active at high temperature, so it is known for keeping it in high temperature, surviving in high temperature. Alright, at 95 degrees temperature, as used in PCR. So this is a basically PCR used enzyme. Alright, so PCR application based enzyme, Taq DNA polymerase. So coming to about PFU polymerase, so what is it all about? So it is purified from hyperthermophilic Archean Pyrococcus furiosus, used to amplify DNA in PCR. This is also a PCR based enzyme. Also it is purified from hyperthermophilic Archean Pyrococcus furiosus. Pardon. So PFU DNA polymerase has superior thermostability. Alright, so let's just mark its function. So it has a superior thermostability and proofreading properties as compared to DNA polymerase 1. Alright, so this is definitely a plus point for PFU polymerase. It has a superior thermostability, provide thermostability, as well as it has a proofreading property or the nuclease activity as compared to DNA polymerase 1. And PFU DNA polymerase 1 possesses 3 to 5 exonuclease activities. So 3 to 5 exonuclease activities from in a reverse direction. So generally DNA strands are in 5 to 3 direction. So this is in the opposite direction. It renders exonuclease activity in 3 to 5 strand, 3 to 5 in 3 to 5 direction. So the exonuclease activity is just removing nucleotides or degradation of nucleotides from the ends, which can be from either from single strand, only from single strand, or from double strands, and that's from the PFU polymerase. So talking about reverse transcriptase, so it is involved in the replication of several kinds of viruses, and reverse transcriptase is unique in that is used in this RNA is used as a template and DNA is not used as a template, because its name suggests, and I hope many of you have heard about this and know this reverse transcriptase, as RNA is used as a template in this, and it is its application is in cDNA cloning. So this is very important, cDNA cloning. I guess many of you know about cDNA. If not, I'll be coming with more such videos about cDNA and other library related function to cDNA. So moving on with so talking about modifying enzymes. All right, so some of the modifying enzymes are alkaline phosphatase. So it is isolated from E. coli and calf intestinal tissue. So it removes phosphate group present at 5 terminus of a DNA molecule. So as you can see, a practical example shown here. Let's say this is a DNA strand as usual. So this has a phosphate group here, and this is generally the 5 to 3 end. All right, so it is present. So as it says, it removes the phosphate group present at the 5 terminus. So this is the 5 terminus and it has a phosphate group. So basically it removes the phosphate group from here. So as you can see here, there is no phosphate group, which is replaced by a hydroxyl, or it is removed by OH. Hydroxyl group is replaced. In the further video I'll show you. So this was the function of alkaline phosphatase. So moving on, some more modifying enzymes which are used in this processes are polynucleotide kinase and terminal deoxynucleotide transferase. So these two are very important enzymes. This is to be noted. So also talking about polynucleotide kinase, it is isolated from E. coli. All right, and it is infected with T4 phage. Alright, so it is isolated from E. coli infected with T4 phage. So this is one term, E. coli infected with T4 phage, and it adds phosphate group onto the free 5 termini of DNA molecule. So it is just the opposite of alkaline phosphatase. So it adds here, adds phosphate group here. Alright, this is the 5 end. Definitely it will add here. Just in the previous case we talked about alkaline phosphatase. This is the opposite of alkaline phosphatase. So to come into the last enzyme, which is the terminal deoxynucleotide transferase. So it is isolated from calf thymus tissue, and it adds one or more deoxyribonucleotides onto the 3 terminus of DNA molecule. So this is the some of the examples shown here. As you can see, this is a strand, this is a single strand DNA, this is a double strand DNA. So its function is to add one or more deoxyribonucleotides onto the three end terminus of DNA molecule. So this is the 3 end, as you can see. This is the 3 end. All right, so it adds this deoxyribonucleotides at here. So as you can see, these are some of the added deoxyribonucleotides at the 3 end, as in the double stranded shown here. So this is the 5 to 3 end, and this is the 3 to 5 end. This is complementary, this is the template, and this is the DNA strand. This is, I hope so you can get this. And from 5 to 3 end, so 3 end, at the 3 end we can add, it adds more deoxyribonucleotides, whereas in the template it is opposite. So this is the 3 end here, so this is added here. So that's it. In this video I tried to explain all of the enzymes which are possible. You can just watch the previous video, which was the continuation. This video was the continuation of the previous video. So you can watch both the videos to get the knowledge of all of the DNA manipulating enzymes that I've talked about. And thank you for watching this video. Stay tuned for more.